Inside of a cell, the nucleus contains the construction plan for life, DNA, which forms the basis of all life functions. Our genetic material is under constant attack from viruses, environmental factors, and bacteria. As a result, the DNA in each cell is damaged 40,000 times per day. It's a problem that humans share with animals and plants. Yet in spite of this, most organisms survive. This scientist has discovered why. The winner of the 2008 Otto Bayer Prize, Professor Thomas Carell, a chemist at Ludwig Maximilian University in Munich. I find scientific puzzles totally irresistible. I have to put all my effort into them, do everything I can to take the project forward, to find a way of solving the puzzle. Martinsried, near Munich, in the year 2004. This is the Silicon Valley of European biotech research. It's where Carell's research into the question of which chemical processes are used to repair the defects in genes entered the decisive phase. He put together a 30-strong team of scientists from different disciplines. Until then, scientists had only found out that proteins are involved in the repair process in some form. But nobody knew how the repair teams bind the proteins to DNA, nor how they identify damage, major damage. Carell chose crystallography as his approach to unraveling the mysteries of these processes. It's a technique in which the enzymes present during the repair are shock frozen or crystallized. X-rays are then used to visualize each single atom. Carell's aim was to gain split-second images of the repair enzymes at work. The biggest problem was that the vital process of repair occurs in the space of 1/billionth of a second. Carell's team began preparing for a very special kind of photo shoot. Thousands of shots would be needed to catch the decisive repair situations and to capture them as images. It's a very labor-intensive method because so many conditions have to be tested first to catch exactly the right moment. I can tell you that it cost quite a few weekends. But that wasn't the only obstacle in this project. Carell's people needed fairly enormous quantities of an exotic starting material in order to observe the proteins during their repair activities. Thousands of strands of DNA with an identical defect were required. The scientists in Martinsried used a synthesis robot and modern software to produce strands of artificial DNA. This is a technique that has only been around for a few years. They then used newly developed chemical methods to damage the DNA in exactly the right way. This is a very laborious process. But perhaps the greatest challenge was the production of the proteins that Carell wanted to use to repair the DNA. This is not something you can simply buy in a supermarket. They had to develop new technologies, including a plant culture system, to obtain quantities like this. And without the new process that we developed, we wouldn't be where we are today. It took 15,000 crystallization cycles before Carell was able to solve the puzzle of the repair processes. And it took an enormous volume of proteins. The scientists ultimately found that yeast and plant cell cultures were able to produce sufficient quantities of the repair enzymes. They used bacteria to introduce the blueprint for the proteins into host cells. These cells then dedicated their entire energy to producing the repair enzymes. Carell's team had overcome all the obstacles and had achieved its objective. 3 images show repair enzymes in the process of repairing damaged DNA. We think the results are sensational because they allow us to observe step by step the way the repair enzymes locate the damage and repair it, or the cell division machine reads over the damage and then produces a mutation. The principle of repairing UV damage functions as follows. The repair enzymes first bend and twist the DNA around its own axis at the site of the damage. The researchers discovered a minor sensation here. It's the blue spectrum of daylight that provides the previously unexplained burst of energy to initiate the process capable of repairing even major DNA damage in a billionth of a second. We were speechless when we discovered that the cell division machine simply kept on running as if nothing had happened. An understanding of these processes could open up new medical possibilities, leading to better treatment of cancer, for example. In early 2008, Professor Thomas Carell was awarded the Otto Bayer Prize, worth 50,000 euros, for his research activities at a ceremony in Berlin. It was presented by Werner Wenning, the chairman of Bayer's Board of Management, and Dr. Wolfgang Plischke, the member of the board responsible for research.